Hi, this is Miss Litton, and this is Chapter 19, Taxonomy, Systematics, and Phylogeny, and this is a continuation from last class. So just a couple things to remember on classification is we reviewed this already at the start, is the categories. So let's say them together. Domain, kingdom, phylum, class, order, family, genus, species. What is the most inclusive category? Domain. Most exclusive? Species. And Linnaeus gives the binomial nomenclature. What does bi mean? Two. So you use the genus species name. Like you are a Homo sapien, so Homo is your genus. Okay, good. And then moving on to where we were talking about a species defined is that they share the same gene pool, that they can interbreed. And we have already had those discussions where we talked about all the different ways you can identify a species. The biological definition is they are interbreeding, right? Okay, we also talked about you could have hybrids. So maybe the F1 is viable, but the F2 is not. So these are some zebroids, which I think are super cool. And then we also talked about subspecies. Now, on the one thing is, did I give you everything through the first box, 19.1? Oh, right, that's right, yay, I forgot I did that. Okay, perfect, so it's just explanations, perfect. Okay, so let's talk about divergence. Okay, so when you see that they both have A, that means they have the same what? Ancestor, right? Okay, so I'm going to let you explain these back and forth to each other, and not it. Yeah, I still don't care. Oldest, you go first. Go ahead. So in divergent evolution, so you do have a common ancestor, and then due to your environment, you are using, let's say, you could have a homologous structure, like when you looked at the arms of a wing and a bat, of the wing of a bat and a human, you could have the same types of anatomy, but you could have a different function. So that's what this is showing right here. Okay, so you have a common ancestor. So we saw this before, right, with these homologous structures. So because they have the same anatomy, you know they're going to have similar what? Because they have the same anatomy. Similar DNA, right? Remember how we looked at the different similarities in developmental biology and how the genes, right, are the same? And so, but they're exploiting different environments. So this is very typical of homologous structures. Okay, now let's look at a different one, and this time it's youngest biobuddy. Talk about convergent evolution. Go ahead.
All right, so let's see convergent.
So you do not have a common ancestor in this one, but you end up with a similar structure. So let's say you have wings. You could be a bee that has wings or a bird that has wings. You're just exploiting your environment in the same way. Or a dolphin with a fin and a salmon with a fin. You just are exploiting your environment the same way. It doesn't mean you have a common ancestor, and these are known as analogous structures. All right? So in this case, and these are examples of analogous structures. So you're just exploiting your environment in the same way. Okay? Back to the oldest biobuddy. Why don't you talk about parallel evolution? Go ahead.
So parallel?
Well?
Parallel evolution. It's coming
Yeah,
out.
but it's not going.
Parallel is
Two same species.
There we go. That was weird. Okay. Here we go. How is parallel, have this discussion, how is parallel like divergent? How is parallel like convergent? Okay, talk about that. So it's like divergent in what? They had a common ancestor. Okay, so they had a common ancestor, but it's like convergent because they ended up with the same structures. Okay, so you can see these when you compare the placental mouse with the marsupial mouse. This is an example of parallel evolution. They ended up in the same way, similar structures, right? Similar environments. Their ancestor was from a long time ago, but they did share a common ancestor. So I made a little chart if that would help you kind of differentiate between those. Is it in your notes, or do you want to pop it into your notes? That might be a good idea. I believe it's back to the youngest bio buddy. Why don't you lead the charge on this chart right here? This should be a review for you. Yeah, the X is what they share, right? The X is the true statement. Okay, so I didn't want to confuse you in this in any way. So homologous structures, do they have a common ancestor? Yeah, and the forelimbs, right, of those animals, that's your common one right there. So common ancestor is a trait found in the ancestor? Yes. And could it have a similar function? Yeah, a bird and a bat have a similar function. A bird and a human don't have a similar function, but they could. They could have a similar function. When they're analogous as a result of convergent evolution, all they have is the similar function. They do not have a common ancestor. That trait is not found in a common ancestor because they don't have one, right? And then parallel evolution, yes, in your ancestral past, you did share it, and you have that similar function as well. Okay, can you differentiate between those three, all right? Okay. Now, next. When you're looking at your phylogenies, you're looking at your ancestral traits. So all of these are related to each other. Okay, so you still have a sheep, a cow, you have reindeer, you have deer, red deer, and then reindeer. So you know reindeer are real, right? That's like a caribou. Did you know that? Yes. Okay. So they all, when you work your way back, right, that you could tell that the sheep is more related to the cow than it is the red deer, okay, when you look at it in this way. So next thing I want to do is I want to go over classification of humans. So when you look at the different identifiers here, we are in the domain Eukarya. Would you have any trouble remembering that? No. Okay. So domain Eukarya, the kingdom that we're in is the animal kingdom, right? Now look at the chart back here. Okay. So if you look, oh, I'm walking too far away from that. If you look at the chart back there, the poster all the way to the left, do you see where it says Archaebacteria and Eubacteria? Okay. Those are those first two domains, right, of prokaryotic cells. Now, when I was in school, that used to be the superkingdom Monera. I doubt you're going to see anything where it says Monera anymore. You might, on a test that you take, but that was when they didn't differentiate between those two domains. Then the four posters to the right, those are all the kingdoms that you would find underneath the domain Eukarya, right? Protista, primarily unicellular, but it could be multicellular. Eukaryotes, fungus, absorptive heterotrophs, plants, autotrophs, and then the animal kingdom. Okay, so that part's easy. Okay, so come back here. Kingdom Animalia. The phylum that we are in is Chordata, and when you're a chordate, you have a dorsal supporting rod and nerve cord. Do you remember when we learned about it in development, the notochord, and how it induced the tissue above to form your neural tube? Yes? And then I said that notochord is going to become your vertebrae. Do you remember all of that? Okay. We are actually a subphylum Vertebrata because there are organisms that are chordates, but they don't have the vertebrae like we do as it develops from that notochord. You just have to have it at some point during your lifetime. You don't have to have it forever. Okay? So we are phylum Chordata. The way I remember that is I think PC as in PC computer or being politically correct. That's how I remember that. Okay? The class we are in is Mammalia. And if you look around the room, look at all the mammals. Now, what makes a mammal? Okay? Mammary glands. And it doesn't matter if you're a boy or a girl, you're set up for mammary glands, right? Everybody has nipples. Right? And the reason why is we've talked about this. When you were developing, your body did not know early on whether it was going to be in the binary world male or female. So the nipples are there just in case, right? But if you are a guy, then a biological boy, then you have the hormone testosterone secreted from, remember, your testes and ovaries came from the exact same tissue, right? But the presence of that Y chromosome and the testosterone then triggered the development. Instead of yours staying as ovaries, they became testes if you're male, right? So that's why you have that. So we all have hair and we all have mammary glands, and that's what mammals have to have. Okay, so I always remember, look around the class at all the mammals. So look away, so see if we can remember them from the beginning. Look away, look towards me. So what domain are we in? Eukarya. So it goes domain, and then it goes what? Kingdom. What kingdom? Phylum. Chordata, because you remember PC, politically correct, or PC computer. Domain, kingdom, don't look, I see you peeking. Domain, kingdom, phylum, class. What class? Class of mammals. Okay, perfect. Okay? Then the order that we belong to is primates, and this is stupid, but I'm going to tell it to you. This is how I remember this. No monkey. Okay, say it. No monkey. Okay? So you're ordering the primate. One more time. No monkey. You're ordering the primate. The family that we are in, okay, Hominidae, because the family is home in the day. Family is home in the day. Okay? Family Hominidae. And then you're golden, because your genus is Homo, and then your species, you have to say both names, Homo sapien. Okay? Now, so let's do it once together, and then you're going to ping pong back and forth. Okay? What domain are we in? Wait, I got to move this so you can't see it. There we go. What? Because I need to be near this. What domain are we in? Eukarya. Domain, then what comes next? Kingdom, Animalia. Domain, kingdom, did, king, Philip, phylum. Phylum, Chordata. Domain, kingdom, phylum, class. Mammalia, because we're all mammals, right? Domain, kingdom, phylum, class, order, and we're going to remember no monkey. So what order are we in? Primate. Domain, kingdom, phylum, class, order, family. Hominidae, because the family is Hominidae. Family, and then it's genus, species. What is it? Homo sapien. Good. Ping pong back and forth with your bio buddy. Youngest, you go first. No. I take it back. Make them turn away. And youngest bio buddy, you go first. You name the classification category, and they got to tell you what man's classification is, and then you'll switch. So turn away so they can't see. And if you're at home, just try to write it down. See if you can remember it.
All right.
Okay, you good? So on your notes, you have the nested hierarchical categories. So you just want to throw man's classification in there so you know it. And you already have the notes on these. These current categories are just the best hypothesis of evolutionary relationships. And you have that. And you also have the highly suggested, bless you, reading and thinking about DNA barcoding, right? How they're trying to do that. All right. Do you need more time, or are you good to go? All right. So on your domains, we have already talked about this. You just want to be able to differentiate between your 3 domains. And I've given you the notes on this. Is it already highlighted for you about comparing RNA sequences that they branched early? Do you have that on your notes?
Yeah.
It's highlighted though? Okay, good. All right, so let's start with the domain bacteria. And why don't we have the oldest bio buddy take the lead on that? You've got the notes for it. You've got 5 characteristics that I've given you on bacteria. Why don't you just ping pong back and forth on those and make sure you understand them. If you're at home, you have your notes on that. Unicellular prokaryotes that reproduce asexually. Most are heterotrophs. Cyanobacteria are large photosynthetic bacteria that were probably the first organisms to contribute to the oxygen revolution. Remember, the oxygen revolution was about 2.3 billion years ago. They are critical in ecosystems because they break down organic nutrients. Some are parasitic and cause disease. Remember, they have circular DNA, no histones. They're not winding the DNA up on histones, and they're very rare to have any kinds of introns. Remember, introns, we cut out the introns before it leaves the nucleus. Yeah, totally. Big review. Okay, did you finish then those 5 items? Was that all review for you? Yeah? We've already learned all of that. Okay, why don't we switch to your other bio buddy and be the expert on archaea. Go ahead. Okay, so for archaea, these are unicellular prokaryotes that reproduce asexually. They live in those really extreme environments like thermoacidophils live where it's really hot, and a low pH. Halophiles live where it's very salty. There are no phototrophs or parasites. Instead, there are some chemoautotrophs. Remember, chemoautotrophs break down inorganic molecules like hydrogen sulfide and use that for their energy. They're different from bacteria because of their rRNA. Their cell membranes have branched lipids. I don't have this in the notes right here, but archaea do not have the cell walls made out of peptidoglycan. The variations in the membrane in the wall probably allows them to function at high temperatures. And they have some introns and some histones like eukaryotes. Okay, come back to me. Do you remember how we talked about, I mean, you could lose a trait that had evolved, but one of the things that makes archaea more closely related to eukarya has to do with what? What did it say in your notes? What do they have? Yeah, the rRNA is more similar, but they have something that bacteria don't. Some histones and some introns. Yeah, some histones and some introns. Also, I noticed I don't have it in the notes, but remember, bacteria have the cell wall made out of peptidoglycan, and archaea does not. Okay? Now, bacteria and archaea, all of them have cell walls for the most part. I mean, there's a few exceptions. Basically, they all have cell walls. But in archaea, their cell walls and their cell membrane, the differences in that allow them to withstand those extreme conditions, you know, allowing them to live where it's really hot, especially in the temperatures, or really salty. Okay, I think that is no photo. You saw that? No phototrophs. Some are chemoautotrophs. If you remember what a chemoautotroph is, they'll break down something like an inorganic molecule like hydrogen sulfide and use the energy from the bonds to synthesize their carbohydrates. If you remember that. Okay, and then your last one is domain eukarya. And I'm going to go ahead and let switch to the other bio buddy. Go ahead and review that real quick. So within the domain eukarya, you basically have 4 kingdoms, depending on what classification scheme you're using. So you have protista, which are primarily unicellular eukaryotes, but there are some that are multicellular. You have fungi, which are basically absorptive heterotrophs. You have plantae, which are autotrophs, and then animalia, which are ingestive heterotrophs. So you have unicellular and multicellular. They all have membrane carbon-bound nucleus. Sexual reproduction, very common, and various types of life cycles. Guys, I would be familiar with the 4 kingdoms that you have within, this is just a chart that shows the 3 domains, but I would be familiar with the 4 kingdoms that we know. Is it here? Yes, it is. Here's a chart for domain Eukarya. You can see this, right, all eukaryotic. They are mostly multicellular. Prokaryotes, usually they're unicellular. There are some multicellular ones, but what their metabolism is, you'll notice kingdom Protista, they're photosynthetic, but they can also be heterotrophic. In Protista, they kind of do it all. There's all different. In fact, you can find like precursors to fungi, plantae, and animalia in kingdom Protista. In fungi, they're absorptive heterotrophs. Means that they're not doing like how we bring food in, we do ingestion, right? And then digest inside. They absorb their nutrients. So I think I've probably mentioned this before, athlete's foot. What's happening is that fungus that's on your foot, it is releasing digestive enzymes and then absorbing the nutrients of your foot, and that might be why it would irritate your foot and kind of feel itchy. I've never had athlete's foot, I tell you if I have. The key thing there, based on what I've already told you today, stop feeling around. But in that situation, you want to keep your socks clean, right? Clean and dry. That's the best thing in order to avoid athlete's foot is if you've been working out and sweating, like say in the morning, change your socks, right? If you work out in the morning, because that can lead to anything kind of growing in that area with that sweat. Yeah. Okay. Kingdom Plantae, they are autotrophic by photosynthesis. There are some exceptions to the rule, like a Venus flytrap or a pitcher plant, but they are usually living in nitrogen deficient soil. So it's not like they're like, I need to go eat. It's just their source of nitrogen. That's an adaptation they have. And then Animalia, heterotrophs, but ingestive heterotrophs, whereas fungi are absorptive heterotrophs. Protista can move by flagella. Fungi are non-motile. Plants are non-motile. We are motile. Mostly sexual reproduction in their life cycles and how that they reproduce. And you could talk about protection of a zygote. You can see that. I don't know that they're going to necessarily ask you about that. All right. So here's some questions. Let's go in today as worst disease we've had. No, it might get gross. It might get gross. Let's talk about favorite milkshake. Oh, you don't like that one. Okay. Pick another one. Favorite. We've already done favorite shoes. Let me just buy you a car. What car would you like me to buy you today? Because I won the lottery, $50 billion. What car? If I said, let's leave class and let's just go get cars for all of you, what would you pick out of the parking lot? And I'm going to say you have to keep it under $100,000 for this car.
I don't know.
Live with it. I want a bike.
With AC.
What car is Marley Anderson?
Lightning McQueen.
Ka-chow.
A school bus. A school bus.
All right, let's go. Here, I'm going to show you the questions. Commit to them at home. We apologize for this class today. So here is question number 1. Pause the video. Pause it, and answer this question. Commit to it. Here's question number 1. Here is question number 2. And here is question number 3. finishing up here. You're almost
Okay,
done.
that's what I thought.
Okay, you ready? Here we go. Which of these are not eukaryotes? Yep, bacteria and archaea. These four down here are all underneath eukaryotes. Really? Who picked this? Fungi, right? So archaea and bacteria those are two domains. Which of these are not eukaryotes? Eukarya, Animalia, Plantae, Fungi. These are the four kingdoms underneath eukarya. Okay, number 2. Which kingdom is mismatched? Fungi, prokaryotic, and single-celled. That's exactly right, because fungi are not prokaryotic. What are they? Eukaryotic. Okay, and then Plantae are not multicellular only. There are some unicellular examples. And D, Animalia, arthropods and humans. Yes, they do contain arthropods and humans. Okay, number 3. Which of these characteristics is shared by bacteria and archaea? They do not have a nucleus because they are by definition what? Prokaryotic, right? So they have absence of a nucleus, presence of ribosomes, absence of membrane-bound organelles because they're prokaryotic, and they do have a cell wall. There are very few exceptions to that. Tell your bio buddy, how did you do on that? All right, moving on to the last bit here. Phylogenetic trees. We already learned how to do this, right? We have learned how to do, and we're looking at the ancestral history. So what these two have in common is that they have the ability to climb trees. That, if you're just comparing these two, that's their ancestral characteristic you could look at, okay, is the ability to climb trees, but these like to run along the branches, and these like to swing from limb to limb. If we broaden it a little bit, we look at an ancestral trait that they all share what? Mammary glands. Okay, so we can keep broadening it back on what your ancestral trait is, okay, in order to get this phylogeny. We already went over cladistics, right? This has already been, we already reviewed that in a previous chapter. Oldest bio buddy, tell them what a character is, please. Okay? And when we look at characters, you might tend to look at just morphology, you know, like this big hangy orange thing hanging over their beak. But it could also be chromosomal or molecular in the sense it could be a protein in the sequence of amino acids in that protein. Switch, other bio buddy, explain cladistics using this diagram, please. Go ahead. And why would you arrange it in that way? Okay, so the bottom two, all of them share A, so that's the ancestral trait. But you're going to group these two in the same one because they have A and C. These, the B, C, D, and E are derived traits, right? Are derived traits. The ancestral trait is what? A, okay? So now, I just wanted to get in a few more specifics about how you do cladistics and set up a cladogram. So, not it. Somebody's going to take the top, somebody's going to take the bottom. Go ahead. This should be review for you.
Ancestral characters, those who are found within the entire line of descent, present in members of one group, but of the line is not a common ancestor.
Okay? Whoever just explained this, other bio buddy, you're up. Go ahead with your cladogram. So you could go back and you could say that this was a clad or clade, or you could go back and say this was a clade, because you're coming from a single ancestor. Does that make sense? And all the derivatives from that. Okay. Whoever was in charge of this page, you're going to be quiet. You're going to do just the middle item on this next page. Okay. So switch the other person. You're going to do 1 and 3, and then whoever did the last page, you're going to do number 2. Go ahead.
So the outgroup is
The outgroup is a species that has more ancestral characteristics than the by sequencing the
when making a cladogram, the primary assumption is that the grade is the number of derived characteristics shared by groups.
The primary assumption, or when making a cladogram, the primary assumption is that the greater number of derived characters shared by groups more recently the groups shared.
Okay? And then the next part, we've already done this. Remember this? And we did this. So that we already know how to do. So just leveling it up a little bit. You could be comparing genes if you wanted to, some sort of biochemical evidence. You could make a cladogram based on the number of mutations if you wanted to look at that, right? The more similar, like if everybody has the same mutation, that might be then that ancestral trait. And then you look to see how many mutations separate them out. When you set up a chart like this one, I set it up, if you recall, it's like, oh, this has everything here. But this one, I remember how we did that one. It was like, I set it up for you. It might be more like this, where you have to go through and say, oh, which organisms have this, which organisms have this. And you do not want to have a trait evolve, de-evolve, and then show back up again, right? So you want to do it it's like Occam's razor, go with the most simple explanation of that. If you hear hoofbeats, assume it's horses, not zebras, right? Some of you are looking for zebras and sheep. You're like, well, it could be sheep. It's probably just horses, right? Go for the most obvious answer of those, right? So you don't overthink something. So if you look here, your ancestral trait is notochord, and then we're starting to drop, we leave out lancelet because everybody else has what? Vertebrae, okay? Then everybody else has lungs. And then these two have amniotic eggs, right? So you want to go with the most simplest explanation of that. We are going to run into a problem, okay? Because according to traditionalists, it's class. We are class what? Mammalia, okay? Now, birds are given their own class, aves. But it doesn't make any sense because they're really closer to what? Reptiles. But traditionalists looked at reptiles, they looked at birds, they're like, they're different, okay? So they gave them their own class, and they really shouldn't have. So polytomy, why doesn't the dark-shirted bio buddy tell them what is poly, just first of all, what does poly mean? Mini, okay. So go ahead, dark-shirted bio, and tell them what polytomies are. Go ahead.
In contrast, any node that only has 2 intermediate descendants.
That's what would be considered unresolved, because it doesn't make any sense, right? That's why birds should be tucked in there with class Reptilia, under that umbrella. It doesn't make any sense. So it should be more like this. Do you see the difference? Switch, other bio buddy talk. How is that different between these two? Here, I'll bring them both dual page. There you go. Okay, so So we're not going to divide into 3 different groups here, right? So technically, according to Cladis, we should just tuck them right in there as part of class Reptilia. Okay, let me go to a single screen. Okay. Next, do you know whose turn it is? Okay, so review a little bit about using fossils in phylogeny. Go ahead.
Using fossils in phylogeny can establish an age of organism, but sometimes difficult to know who's related.
Right? We've talked about that, right? Because you can't look at fossils and know their behavior. You can't understand their sexual selection, mating preferences. You can't tell that in fossils. The fossil record is incomplete. Where are you most likely to see your best fossils? In what kind of rock? Sedimentary rock, right? But you have to have a tissue that can be fossilized, and then it has to be fossilized and then found by a human, right? So that's not always going to happen. Other issues, you can look at just morphological traits. Switch, whoever's turn it is. Should be a good little review for you.
But then you have to be careful of analogous structures because they're the results of their divergent
And so morphology, you got to be careful when you're looking at morphological structures. You cannot tell necessarily the results of convergent evolution, right? You wouldn't be able to tell necessarily. Okay, and switch one more time, looking at behavioral traits. Okay, and then we said let's look at the molecular clock. We can look at things like mutational rate, right? We can look at similarities in proteins, similarities in DNA. Whoever did behavioral traits, you're doing this one too because that one was too easy. Go ahead, do this one. What group is primate? What classification group is primate? Order. No monkey, right? That's the classification category of an order, right? Do you remember that? Okay. And then switch one more time. Go ahead. We've seen this before as evidence of evolution. Right? And switch one more time. So what mutates faster, and I'm using the term loosely and in air quotes, what mutates faster, whole wheat bread or Twinkies? Whole wheat bread is going to degrade faster than Twinkies. Do you agree with me on that? Yeah. So think of Twinkies as like the nuclear DNA, right? So for closely related species, if you were looking at the nuclear DNA, it's like looking at 2 Twinkies. It takes them a long time to degrade, right? So then move out to the mitochondrial DNA. Compare the bread, right? Because the bread is going to mutate. It's going to mutate faster, right? It's going to break down faster. So that's going to help you with closely related species to focus on the mitochondrial DNA because the change will happen quicker. All right? Here are some more questions for you. Or maybe one question, just one question. Yes, one question. And here's that one question. Have everybody? I think we do. I end you. Yay! proud of you. The end. Okay, and I'm not going to stop the video because I'm going to bring up the debrief so we can make sure we're good on that as we prepare for our test. When is our test? Monday. Awesome.
I'm so excited. So excited.
Do you see it anywhere? Yes. All right. So, did you get a chance to look at your last multiple choice? No. So today, 7th period. I think I'm open help for honors today. So you can look at the multiple choice today at 7th period. Tomorrow, remember, I'm off campus again. So if you want to look at that, it's got to be today. All right. So on your Unit 7 AP debriefs, here we go. Idea of natural selection. Could you explain to me what natural selection is? Yes? What are the premises of natural selection? This is kind of like your review, since we're not going to have another review, right? Because your test is Monday and I'm not here tomorrow, right? Okay. So what are the premises of natural selection? Survival of the fittest, okay. But let's lay it down. First of all, we have overproduction of young, right? What? Yes, we're going to have variation among those. There's going to be competition. Not all are going to survive. Some of those variations are due to, what did you say?
Mutation.
Yeah, genetic variations, right? And those that are best adapted, remember we use the word fitness, right? That have the ability to pass on their genes. Those traits that are selected for are going to be passed on, and that you have vast amounts of what? Time. Vast amounts of time. We talk about selection. All selection boils down to your ability to get your DNA passed down, right? When we talked about different kinds of selection. Directional selection is going towards one extreme, right? Disruptive is both extremes. Stabilizing is the norm. But some of these selection pressures can lead to speciation, right? So we kind of just tying that in there right now. And then sexual selection is anything related to facilitating your fitness, right? That's sexual selection, is facilitating your fitness. And it could be intrasexual or intersexual. Do you remember that? Intra would be like what? Males competing with males, right? No? Yes. Inter is male and female. Good. All right. So idea of natural selection. Evidence of evolution. Name, description, and authentic example. Right? So what were the pieces of evidence of evolution? Remember that chart and I would pull them out? Fossils. Boom. So with fossils, you can bring in the whole idea of how we date fossils, right? What are the 2 ways we can date fossils? Relative. Perfect. Okay. We can talk about index fossils, right? We could talk about the geologic time scale, Cenozoic, Mesozoic, Paleozoic, and Precambrian. We can bring that all into fossils. We could talk about transitional fossils. We can talk about the rate that we see, punctuated equilibrium versus phyletic gradualism. Which one would you see more fossils in? Gradualism. You would see like a lineage, a change. What is the really good example of phyletic gradualism that we used? Horses. Right. Okay. Good. So evidence of evolution, fossils. We can bring so much into that. What's another piece of evidence? Biochemical. And that's the big one today, right? Comparing DNA, protein, comparing molecules. Similarity in that, right? When you look at similarities of things like, what was that? Cellular respiration, right? When we look at things like the genetic code, the universalness of the genetic code, right? ATP, DNA, yes? Okay, give me another piece of evidence. Anatomical. Anatomical, we had 3. What were the 3 anatomical pieces of evidence, and what is definitely shut the door on? First, tell me who you're shutting the door on. Right, because that means just similar in function. Who do we welcome? Homologous structure. Same anatomy, probably similar DNA. Could be used for the same purpose or not. What's the next one? Vestigial structures. This points to our ancestral past and our common ancestry. Could have been useful in our past, not useful now. It shows where we came from. And what's the third one? Embryological. Okay, then another whole category was development, and that's looking at those master switches and those developmental genes, right? Now you can look at embryological development. You look at actually how the baby develops, right? You could look at, so I don't want to confuse those two, but that's a piece of anatomical evidence, is the physical structure of development, and we can see kind of evolution on fast forward, right? And when we see it go through the different structures, and then similarities between embryos. Then there were developmental genes that we looked at, and the similarities in those. What's another piece of evidence? Biogeographical. So then we're looking at continental drift, right? When were the continents together? We're looking at adaptiveness, how it can change over time depending on where the continent is. Looking at fossils in Antarctica of reptiles, where we know now in Antarctica you couldn't survive, but that must tell us that at some point it was adaptive and was in a different area. We look at the fern, Glossopteris, I believe it is, where it's uniformity amongst the continents, right? We look at mammals and the divergence. Good. Do we hit them all? We did. Chapter 16. What are the 5 factors that can impact allele frequency? Let's do the 5 factors. What's the pinky? Small population size, non-random mating, mutations. What did you say? Gene flow, natural selection. Those are the 5 factor. What is the Hardy-Weinberg equilibrium and how does it relate to evolution? The idea is in Hardy-Weinberg equilibrium that the allele frequencies will stay the same. Okay? How do you make them the same? You do the opposite of the 5 things that can change it. So instead of having a small population, you would have what kind of mating? Random mating. You would have no. You would have no. I mean, you would have no gene flow because you're concerned about genetic drift. And genetic drift is all about, I'm throwing dice. Chance. Good. And you would have no what? Yeah, no selection. On the genetic drift, what were the 2 types of genetic drift? Bottleneck, which means you had a disaster, and then the other one is founder's effect, where you start a new colony somewhere else. Yes? Okay. Name and describe 5 different types of what? Name and describe the different types of evolution. Oh, we already went over those. Directional, right? What else? Stabilizing, disruptive, and sexual. Okay, good. All right, we good on that one? So far seems pretty straightforward, right? Wait, go ahead. I'm listening. What did I miss?
I've got micro and macro.
C in there.
C is micro and macro.
Wait, tell me. I can't hear. To one person.
Micro and macro.
Oh, micro and macro evolution. Yeah, I should probably differentiate that. Micro, what was microevolution? Small changes within the species. Good. And macro is? And let's talk about types of speciation. Is that in this one or the next one? Yeah, it's in this next one. Okay, good. All right, so 17, name, describe, and give an authentic example of reproductive isolating mechanisms to prevent gene flow between species. Let's name them. There's like 10 of them, right? What was the first one? Well, there's pre-zygotic and post-zygotic, but underneath pre-zygotic, what was the first one? Habitat. So we live in different places in our habitat. What was next? Well, so we reproduce at different times. What was the third one? Behavioral. So you're meeting the first 2, you just don't know each other's dance. What's the next one? Mechanical. Our parts don't fit together. What's the next one? Gametes. So the gametes, you might not have a receptor. Now we're going post-zygotic. What was the next one? Hybrid inviability. So it doesn't develop. Or you have fitness issues, right? Either in the F1 or in the F2. Yes?
So the behavioral, it just means that you don't act the same? Or?
We don't communicate that we want to have reproduction. You're not doing the dance that I need. You're not behaving in a way that makes me want to reproduce. Maybe our parts would fit together, our gametes would fit together, but because you're not dancing correctly, I'm not going to mate with you. Okay. B. Describe and provide examples of the following forms of speciation. Allopatric. Give me one word. Barrier. That was what I was thinking about. Yeah, that's 2 words. But barrier. And that barrier is going to be more something in, like she says, geographical barrier. It's going to be something that separates the 2 of you. And you have your own selection pressures, your own initial gene frequencies, right? Your own mutations. So over time, when you're rejoined together, you no longer want to interbreed. That is the most common form of speciation. Sympatric speciation, you were standing right next to me and you changed. More, it could be due to habitat preferences or food preferences, but more than likely it's due to what? Chromosomal changes, right? Remember, auto and allo polyploidy. Which one was two different species? Allo. Like I see the two L's and I think two different species. Okay? Allopolyploidy is I made a haploid gamete, you made a haploid gamete, we fertilized each other. We are not diploid because we're two different chromosome sets, but we underwent mitosis, now all of a sudden we're diploid. Okay? Whereas autopolyploidy was I didn't do what correctly? I didn't do meiosis correctly, and as a result of that, I'm still diploid, right? And my diploid got together with your diploid. Now we're quadruploid, or whatever we are. We're polygenic at this point. Who does this usually work for? Plants or animals? Yeah, you're not going to find this in animals. Okay? Adaptive radiation. What's a good example of adaptive radiation? Finches. And this also relates to the value of extinction, right? Or sorry. Yeah, that's what I wanted to say. That sounds terrible. But when you have an extinction event, it leaves room for more adaptive radiation of upcoming species. Okay, what are the two models of evolution relating to pace? I already think I went over these. What are they? Gradualism and punctuated equilibrium. Okay? Look at us reviewing for your whole unit test. Chapter 18, name and describe the key steps of chemical and biological evolution, i.e. what happened and how could it have happened. So, first of all, we have the chemical evolution and then the biological. And I went over that yesterday in a full review, yes? So you have to form, you have inorganic gases, you have to form the monomers. From the monomers, you have to form the polymers, and then the protocell, and then the self-replicating cell, right? And so you want to review the steps and processes of that. Name the major time periods and eras. We already went over that, right? So you can watch that review from yesterday. What role does extinction play in evolution, and what role does it play? Leaves room for others. I'm sorry, I posted this and I thought I did talk about chapter 19. Describe phylogeny. Do you want me to do this? I'll type it right now. And I'll have to repost it. Is it on the classroom one? Yeah. Oh, good. Okay. Because I think I talked about the process of phylogeny. Yes? I'll bring it up so that we have it. Yes. I can't hear. Let me answer that question. Say it again.
For the test, is the multiple choice on all the chapters?
Yes.
And then is the essay on all of them also?
Yes, it is. So chapter 19, just to get this one, describe our system of classification. So that's your domain, kingdom, phylum, class, order, family, genus, species, and no man. Name and describe the three domains. That should be a review. And what is phylogeny and how can it be determined?
So the highest choice in the reading, but we can also ever.
Highly suggested reading is all up for game. All the notes, remember? The essays on everything. Essays on everything. And that is it. I hope that was helpful.